Hello, everyone. My name is Vedant Matabushi, and I'm a rising senior at Mission San Jose High School in Fremont, California. Today, I have a special guest with me from Stanford to discuss an up-and-coming field of cancer research that is completely altering the way that we think about oncology. Please welcome Alexander Hankala, who also goes by Xander. Thank you for joining me today.
No problem. Glad to be here.
So, tell us about yourself and please give us a short introduction to the field of cancer immunotherapy.
Sure. So my name is Alexander Honkala. I went to University of Michigan for undergraduate. I've done immunology research, started companies, started nonprofits, and have been also a grant writer and program developer in many contexts. I currently do that with the Xander Laboratory, and I'm also doing a computational biology PhD at Oregon Health and Sciences University. So as to the field of cancer immunotherapy, trying to figure out how to give a short introduction to it because it's one of those fields that's complicated and warrants a long introduction in many ways. What it comes down to effectively is the recognition that we can use the immune system to fight cancer. And with the increased power of post-genomics technologies, we've come to understand that there are endogenous processes going on whereby the immune system surveils and fights against cancer development. And in doing so, tries to eliminate it before it becomes a clinical concern. And that cancer, in order to become a clinical concern, has to subvert some of those processes, and those processes are promising candidates for therapeutic modulation. And we've already seen early success by beginning to tweak the immune system to change the tumor microenvironment, which are specific immune cells around it, that then provoke specific anti-cancer immune effects. And the more we do that, I think the more we'll understand that there's going to be variation between patients, between tumor sites, and even between time points that be taken into account for what strategy we apply to modulate tumor microenvironment immunology in provoking therapeutic effects.
Interesting. All right, so how has your academic career led you to your current research subjects and interests?
Sure. So my academic career has been a little bit shotgun. I did host pathogen interaction research as an undergraduate at University of Michigan. And that's where I first kind of discovered that immunology was really complicated and cool. And ever since then, I've been fascinated. It's a complex dynamic system that's involved in every process in our body, from fighting off disease to maintaining homeostasis. It's involved in the development of our bodies as well as the aging and degradation thereof. So I kind of regard now the body as a bonsai that the immune system is taking care of and shaping over time. And with every new discovery we make, the immune system seems to get more complicated and more interesting. Like earlier today, I was watching a really interesting talk, and I hadn't even known that there was such a thing as peripheral neuroglial cells. Okay, I knew that there were glial cells in the brain, but I was under the impression that that was a yolk sac monocyte-derived restricted lineage that was sealed behind the blood-brain barrier. But it turns out, okay, we have these glial cells and peripheral nerves as well, and that leads immediately to the question of what's the difference between that glial cell and the tumors in the macrophage. And right now, we don't know. There's a lot of open questions. And the kinds of questions that we can answer are only getting more fascinating as we get more technological power, from single cell omics to spatial omics, to even kinds like dynamic synthetic biomarker measurements.
That brings me to my next question. It's very relevant to what you're discussing. What excites you the most about cancer immunotherapy?
So there are a couple ways to answer that. The first thing is that this is a necessary part of making cancer a curable disease in most cases. Given that we now understand that cancer varies so much between individuals and between sites within that individual and between time points and after treatments, and all these other sources of variation, it changes the strategy. There's not going to be some silver bullet new chemotherapy that knocks out most cancers. There's not going to be a specific molecularly targeted therapeutic that works for 50% of cancers unless we can somehow drug MYC. And that might happen down the line. So where this gets interesting is we're going to need a bigger and bigger arsenal of both companion diagnostic biomarkers to define what makes a given patient's cancer different, as well as how that relates to therapeutic options to then treat that patient at that time point for the greatest efficacy. So it's becoming more personalized, it's becoming more complicated, and cancer immunotherapy is a necessary part of that. At the same time, as the second part of that answer, classical chemotherapy and radiotherapy also has immune effects that are dose dependent. And then if you give a relatively smaller dose like metronomic chemotherapy, you see greater activation of CD8+ T cells. But if you give MTD-based chemotherapy, that might be too high, you can actually kill some T cells off and need a little bit of immunosuppression. So now we're saying we have to balance even the use of traditional chemotherapies or even new chemotherapies with their immune effects. And then we're also seeing, even the molecular target therapeutics like vemurafenib, that there are peripheral immune effects that also matter. But each of these things are also things that we can monitor to see how disease is responding to treatment over time, and in doing so, make it more adaptable. Because something that just drives me nuts in how we treat cancer right now is we wait for radiographic progression, in most cases, to alter treatment courses. And when we're waiting for radiographic progression, what we're doing is we're saying, okay, hey, treatment A is eliminating all the sensitive cancer cells. Great, maybe it's 90%. But then that 10% looks like, regression at first, but then we keep giving that treatment and that 10% becomes resistant. And then when it grows back to look like it's progression under radiographic imaging schema, we have allowed the tumor to become recomposed with resistant cells. So why aren't we changing treatment earlier based on biomarkers actually telling us something about the cancer biology underlying it? And cancer immunotherapy, by dint of immune trafficking into and out of the tumor microenvironment, is I think a prime candidate for identifying those kinds of biomarkers for modifying treatment over time. Like if you look at the work of Matthew Spitzer recently, he's been showing that it's not just the primary tumor microenvironment that mediates immunotherapy effects. You also need secondary immune environments activation, like CD8 T cell priming in peripheral lymph nodes that then traffic to the tumor microenvironment and do something before becoming exhausted by local immunosuppression. So that's 1 and 2 answers. The third answer of why I'm excited is cancer research in a way is sometimes 3 to 5 years ahead of the disease areas. And we're recognizing that cancer is extremely personal, but we're beginning to recognize that things like Parkinson's or multiple sclerosis or even rheumatoid arthritis are also personalized. There's also patient heterogeneity in those diseases, and we also need to use immune modulatory treatments in those diseases to get better treatment effects over time. And we're seeing some of that happen. Things like Remicade being on the market for RA for a long time have proven the proof of concept that that will work, but whether or not we should be giving it at a steady state or instead trying to change the immune system to be transiently inflamed to then reset itself out of that pathogenic state remains to be seen.
That's a very interesting point. All right, so next up, are there any discoveries that you have right now that you're proud about and you'd like to share with us?
That is a great question. And unfortunately, right now, I think the answer is no. Simply because the most recent findings are all preliminary, and I'm in the middle of them. So like I'm acutely aware of how limited they are and how much more work needs to be done before something I can really share and say like, yes, this is true, or this is actually something that's clinically viable. So unfortunately, on this one, I think I have to pass for the moment.
Oh, okay, I see. But are there any discoveries that you've made so far that have been actually clinically proven that you believe have some potential in the same sense that, like I'm saying, like something that you're proud of that has already been proven by?
So I think with that, we could go back to the MDSC nanoparticle project, which the goal was to develop a nanomedicine out of previously approved components to make it as simple as possible. And in doing so, try to get preferential accumulation of metabolism-altering drugs in immunosuppressive macrophages associated with breast cancer progression. And with that, over a couple of years, we reduced the concept to proof of practice and show that with putting effectively Neupogen on the surface of albumin nanoparticles, we could get these to preferentially accumulate in what's called myeloid-derived suppressor cells in the breast cancer tumor microenvironment. And that was just proof of concept in mice. But it's also worth noting that this same hypothesis has also been clinically validated with work by Incyte with their IDO inhibitor. Even though that one specifically failed, there is still a lot of excitement for doing MDSC modulating therapies like Plexicon's work with the CSF1R antibodies or other drugs. May not be an antibody at this point.
So how do you envision this field will look like in 5 to 10 years?
So I think in 5 to 10 years, what we'll see is a greater arsenal of drugs on the market. There's been a lot of Me Too drugs as well with follow-on anti-PD-L1 inhibitors. But we also have identified a big handful of other targets that may also make sense as checkpoint inhibitors like LAG3 or TIM3, while we're also trying to work on things like allogeneic NK cell therapy, macrophage conditioning, and CAR T for solid tumors. I think on the 5 to 10 year timeline, we'll see all these modalities approved for cancer and making significant impacts on multiple different types of cancer, whether that's melanoma or liquid cancers or even solid cancers. The other part of that I think is really exciting is work that's being done with intravital imaging. So intravital microscopy is by which you can you put an optical window into an organ, and then you can fortunately label immune cells or drug agents and use confocal microscopy to see how it's functioning in its native environment. And that's being increasingly used for testing various things like the Caragiannis group tested robastamin, which is an angiopoietin receptor inhibitor, to see whether that could inhibit the function of tumor-associated macrophages in educating pre-metastatic cells as they migrate out of the metastatic niche and go into the bloodstream. So if we can use those kinds of techniques coupled with downstream spatial omics, we're going to understand so much more about both cancer biology as well as immune responses to it. And I think that's going to kind of come down to seeing that there's a bunch of small niches within the tumor microenvironment, some of which are tertiary lymphoid organoids, some of which are perivascular niches, some of which are other niches yet to be identified, and that those niches will constitute functional units that themselves need to be modified by the immunomodulatory therapies that we're trying to use for a therapeutic benefit down the line. Another point I think is exciting is the use of organoids as preclinical models, because traditionally we just put immune cells in medium. Sometimes it's attached, sometimes it's liquid, but these are not very representative of their context in vivo, because we're now understanding that tissue-resident macrophages are very different from bone marrow-derived macrophages, and an organoid gives you the ability to have both the stromal cells, the functional cells, the cancer cells, and the immune cells all together in a small, reproducible, experimentally tractable format that you can use for a variety of different post-genomic sequencing techniques or imaging techniques to find out more about the biology. So all of that's exciting. So a point I mentioned earlier about there being immune phenotyping in patients, I think is also really exciting. So like Personalis was spun out of Dr. Xander's laboratory a while back under the idea of using high dimensional immune phenotyping in patients' peripheral blood cells to say, okay, this patient will benefit from anti-PD-L1, this patient will benefit from anti-CTLA4, this patient has this mutation and needs this kind of drug. And that kind of actual patient stratification is going to become more and more urgent as you go forward, because just throwing everybody on first-line chemotherapy is not, a viable strategy for half the patients that it fails in.
So for those who are unaware, how does omics play a role in cancer immunotherapy specifically?
So omics plays a great role in understanding mechanisms of action. And that's a really loaded term in all of molecular biology, because when you are trying to prove mechanisms of action, it's going to be method dependent. So canonically, before omics, the way that we would do that is we would engineer either a mouse line or a cell line to overexpress a gene or knock out a gene and see if it met the criteria of being sufficient and necessary for a given phenotype. But that was really limited. With omics, we can get a much better kind of idea of what's going on at a global view. So if we're doing something like RNA-seq for transcriptomics, we get an idea of how is a given drug affecting internal cell signaling. If we use spatial transcriptomics, we get an idea of how cells are different and how they're interacting with neighbors from neighbors or in cell neighborhoods over time to see what's the spatial organization of cells in their organization. Proteomics gives us an idea of what's going on at the protein level, because if it's in RNA, it doesn't mean that it's going to get translated into protein. And then you also have differences in how long a protein lives or how it's modified over time. And it's just giving us a much more complete picture of biology, because traditionally we can only look at, like, say, 15 years ago, we had to do maybe 3-4 channel flow cytometry. You are not getting very good cell phenotypes. You're saying, okay, this is CD8 positive, CD69 positive, great. Tells you just a little bit about it. But now that we can measure so many different markers, we can tell a lot more about the actual functional phenotype of these cells, and in doing so, test multiple hypotheses at once, although we be careful in how we define them. It can also generate data sets that go beyond the immediate question at hand. So things like TCGA, having spatial, it's not spatial yet, but having transcriptomics for so many tumor samples has let us figure out what's the global pattern of mutators driver genes, passenger genes, chromosomal rearrangements across all kinds of human cancers. And in doing so, see the development of the next generation of cancer drugs. And now we're getting spatial transcriptomics, human tumor atlas network, and that's going to, I think, drive the next generation of cancer immunotherapies as well. So omics is kind of like, actually, let me back up a little bit. There's a metaphor that people have used a lot in molecular biology of that you have to formulate a hypothesis to find your keys under a street lamp at night. But if your keys are outside that street lamp, you're not going to find them at night. And traditionally, that street lamp is a really narrow piece of illumination. So be careful about your hypothesis to be inside that and make sure it's answerable. If your keys are outside that cone of light, you're never going to find it. But with spatial omics or omics in general, we broaden that light cone substantially and illuminate a much bigger area. So we can address not just the question at hand, but also other questions down the line and generate data sets that help not just the researcher that generated that data set, but that also helps other researchers and other laboratories sharpen their hypotheses, find preliminary sources of validation. And there's also now, I think, the rise of laboratories that specialize in interrogating data sets that other people have made.
So looking back on your life so far, if you had the opportunity to change anything on your path towards where you are now, do you take it? And what will you change?
Let's see. There's a difference between perseverance and stubbornness that is sometimes subtle. And there is the phrase that sometimes people in Hollywood use where you have to kill your darlings, where you have to be kind of a jerk about your own work and your time commitments. And then you have to recognize that something that you're working on isn't panning out. And, you know, sometimes you start with best intentions. It leads to good connections. You get some progress going, but it stalls out for whatever reason. And it's important to be able to detach your ego or your previous time investment from that work and step back and actually quickly evaluate, should I keep working on this, or has this done all it can and it's now time to move on to something else? And there have been a couple of cases where I have probably stuck on to projects longer than I should have, where I'd done initial good work, but kept trying to hack at it for just too long. And sometimes it was a matter of data. Sometimes it was a matter of people I was working with. Sometimes it was a matter of there just not being the resources ready for that kind of hypothesis that was necessary for the next step. So you have to be willing to kind of take that step back and depersonalize yourself from your own work and evaluate, is this the right thing to keep investing my time in or not? But at the same time, that's a hard skill to cultivate, because as you do this, you get more perspective and can more readily recognize when something isn't working out. So that's something where I think biology can learn a little bit from tech industry of trying to fail fast. Design an experiment that validates whether this is worth doing, and then use those results to make that decision, rather than hanging on to it with multiple different areas of work. Like, let's say you're chasing multiple lines of grants or multiple lines of funding, or trying to find the right team for it, but it's not kind of getting to the next step. Sometimes your time is better spent elsewhere.
That's a very interesting point to make. We can all learn from that. So moving on to my final question, do you have any advice for high school students or college students looking to go into this field of cancer immunotherapy in the field? I mean, it's a very promising, and from the points that you've given so far, I'm very intrigued by it. I'm hoping to enter that as well.
Okay. I think the primary advice I would have would develop a note system. I did not have a note system when I first got into science. So like the papers that I read when I was first getting into host pathogen interaction studies, I was literally printing them out, stapling them together, and marking them up with a pen and paper. And I've since moved like a dozen times. So like I no longer have those stacks of paper. I moved across the country. I'm not bringing them with me. So having a note system that is electronic, backed up, and resilient to how you like to take notes and then read notes later on will help you kind of capture value as you keep working in this field and learning more and more about it. Because it's something you can look back to and refer to over time. And we're also seeing, I think, a new wave of note-taking apps that go beyond traditional static notes. Like Evernote and Apple Notes are great for acting like Post-its, but they're not good for like searching your notes over time or making them together or having rich sources. So tools like Canopy or Rome Research or Obsidian are now out there and let you link your notes together and learn over time to build mental models of the systems that you're working on and be able to support those is incredibly valuable, because like as you move forward in your career, you're never going to have a completely perfect model of like what a T regulatory cell is doing, say in melanoma. There are just way too many details. It's too deep a hole for any one person to go down and understand absolutely everything about anymore. So you're going to have lenses into that system that each constitute a mental model. And being able to have multiple mental models is one of those things that differentiates great researchers from okay researchers. So having the note system to then back up those mental models is only going to help you accelerate that process of creating those models and using them in your work, whether that's writing a grant, writing a paper, justifying experiments, interpreting results, whatever it is.
Thank you, Xander, for joining us today. Your passion about cancer immunotherapy is very inspiring, and I'm sure that a lot of people will find the inspiration that they need to enter the field that they find interesting as well. This was very enlightening, and like I said again, I wanted to thank you for being able to join me today.
You're welcome.
So
I can help.
I would like to personally thank the Snyder Lab, as well as Jennifer Lafreniere and Christian Cha, for this opportunity to improve my understanding in the biological and health sciences. Thank you for watching. If you haven't already, reading the accompanying article for this interview would also be a great step to take.